Hello, my name is Juliet Cifuentes, and I will be introducing the cell cycle. I will be speaking about the beginning stage of the cell cycle, which is the interphase. The cell cycle is a series of growth and development steps a cell undergoes between its formation and reproduction. In eukaryotic cells, the cell cycle is divided into two major phases, interphase and mitosis. Interphase is the longest part of the cell cycle. This is where the cell grows and copies its DNA before moving into mitosis. During mitosis, chromosomes will align, separate, and move into new daughter cells. The prefix inter means between, so interphase takes place between one meiotic phase and the next. Interphase consists of 3 steps. The first step is the G1 step. This is the first gap phase. The cell grows larger and organelles are copied. The next phase is the S phase. S stands for synthesis. In this phase, the cell synthesizes a complete copy of the DNA in its nucleus. The last and final step is the G2 phase. This second gap phase is where the cell grows more and makes proteins and organelles. This is also the phase where it begins to recognize its contents in preparation for mitosis. Cells that are meant to divide will complete G2 and enter mitosis. Other type of cells that divide slowly or not at all may exit the G1 phase and enter a non-dividing state called G0. Some cells remain here indefinitely, while others may re-enter division under the right conditions.
My name is Lucia Mestis Nunez, and I'm going to be talking about the mitosis stage called prophase. This is the first stage of mitosis that occurs after interphase. This is when the cell begins breaking down some of its structures and starts building other ones, which also sets the stage for chromosome division. Usually, the stage can be broken down into early prophase and late prophase, which is also called prometaphase. During the early part of prophase, the mitotic spindle forms and the chromosomes begin to condense. This spindle is made of strong fibers called microtubules. This spindle organizes the chromosomes and moves them around. This spindle is also growing between centrosomes as they're moving apart. During this time, the nucleolus disappears, which is a sign that the nucleus is getting ready to break down. During the late prophase part of the stage, the spindle actually starts capturing and organizing the chromosomes, which have finished condensing, so they're super compact. The nuclear envelope, which is a membrane barrier that separates the nucleus from the cytoplasm in eukaryotic cells, also starts to break down, which releases the chromosomes. As the mitotic spindle begins to grow more, some of the microtubules start to capture the chromosomes.
Oh, hello. I'm Angelina Abraham. Continuing our discussion and diving deeper into the cell cycle, the next phase is metaphase. In this phase, the centrosomes are now on opposite ends of the cell. The nuclear membrane is now gone, and the most integral part is that the chromosomes are now aligned in the middle of the cell. So spindle fibers or kinetochore microtubules, they now, from either ends of the poles, they now attach to the kinetochores of the chromosomes. Essentially, they pull the sister chromatid back and forth until they are centrally aligned in the equator of the cell, otherwise known as the equatorial plane. An important part of metaphase is the metaphase checkpoint. This essentially ensures that the cell is ready to divide. It makes sure that the chromosomes are properly aligned and that the kinetochores are properly secure. And if everything is in order, the cell is now ready for the next phase. Take it away, Rachel.
Hi, I'm Rachel Steiner, and I'll be talking about the third phase in mitosis. The third phase in mitosis is anaphase. An anaphase is when the sister chromatids begin to separate and move to opposite ends of the cell. So basically what's happening inside the cell is the proteins begin to break down the sister chromatids and they separate to opposite ends of the cell, creating 2 whole chromosomes. There's also microtubules within the cell, and those begin to elongate with this separation, creating a very long ovular shaped cell. I have a diagram here that shows you the microtubules, which would be these gray lines, and you can see the shape of the cell as it's beginning to divide. This entire process of anaphase is fueled by motor proteins, and that just allows the separation. During anaphase, cytokinesis can also happen. And cytokinesis is the separation of the cytoplasm and the outer layer of the cell. And this is a more physical aspect of the division. And so when you look at a cell that is in cytokinesis, you can see that they're still together at the beginning of anaphase, but they're getting thinner and thinner in the middle because they are going to separate. So that's all for anaphase. On to telophase now.
Hey, my name's Abdul, and today I'll be talking about the fourth phase of mitosis, also known as telophase.
To better explain the process of telophase, let's take it to the drawing board. Before we actually get to the diagram, one of the most important things to happen in telophase is actually that the chromosomes that first start to appear in prophase start retracting and unraveling at the beginning of telophase. Directly after anaphase, the cleavage furrow, as you can see pictured here, separates the two daughter cells, which are inside here and here, which will further allow them to enter cytokinesis. The nuclear membrane that starts dissolving in prophase now starts to reform, except now, instead of there being one membrane, there's going to be two membranes, each surrounding the two future daughter cells. As you can see, the spindle fibers here and here begin to depolymerize and turn to a monomer, which create these nuclear envelopes surrounding the chromosomes. And later, they'll become the nucleus. And this concludes telophase. Now, moving on to cytokinesis.